Hi, my name's Dennis. So what we're doing here is we're finishing up the DNA extraction that you just saw. So what happened was we did the overnight DNA digest for the slug tissue. And now we have these tubes here, and you can kind of see they're a little dark and funky looking. And by the end, they should be nice and clear. And all this dark stuff and a bunch of other things you can't see in there are the things we're trying to remove so that we only have the DNA left. So to do that, we have this nice protocol here, and it involves washing is the term we use, washing the tubes and filtering them in these little miniature filter columns. The filter columns are two parts. One, the actual filter, and then the collection tube. And this will be our final clean one. And then at the very end, we'll have them placed into their individual labeled DNA tubes. So this will be only pure DNA once it's in here. Go ahead and get started. So the first buffer we use is buffer AL, which is this guy. And we take one of these things over here called a pipette, or in particular a micropipette or a pipette man. For most science and most things in this lab at least, we have a detailed protocol about what to do so that everything's standardized. So you always have your protocol available to you, and they're laminated so they don't get wet or anything like that. And so you'll see me referring to that many times, just double checking, because there are a lot of steps and can easily get mixed up if you aren't being careful. So our pipette man is used for measuring out very small quantities of water, or any liquid really, and then pipetting them or moving them. So what I'll be doing is I'll be taking 200 microliters of this buffer and putting it into each of our tissue tubes. Grab our 200 microliters, and there we go. Now, the tips are disposable, and you use a new tip for each sample to prevent any kind of cross-contamination, because we're trying to look at each of these individually. Take our buffer, insert it, draw our tip. Right, and so now that that buffer AL has been added, we actually have to let it sit for just a little bit, only for a couple of minutes. And so after this, we start adding in more buffers and things like that. Now that we added the buffer, we go ahead and do what's called mixing by inversion, and that just means flipping it upside down slowly and then flipping it back the other way slowly. If you shake it a whole lot, you'll end up damaging the DNA. So that's why we do shaking by inversion. And so normally I'll have a full rack of these, and it'll be more than just 5. And so you'll take it and you'll put your hand on the whole thing and flip it slowly and then flip it back, inverting it every 3 minutes, well, for a 10-minute period. So doing it 3 times over 10 minutes while the buffer does its digestion process, the lysis buffer. So depending on the tissue type and what we're digesting. So for this, it's slug tissue and a couple different types of slug. So when you saw them before, they looked all more kind of like this. Although this one, if you look closely, it's kind of more turbid. It has all those white flecks in it. And there's this one, which is very clearly has a lot more white flecks, and just a lot of junk in it. So it's not 100% this will happen. It depends on what part of the slug got snipped and a lot of different factors. So when this does happen, we have to reheat the samples. And so we'll go ahead and stick these back in the incubator for another 10 minutes or so till these have dissipated. Yeah. Now we wait. So now that they've been inverted, we're going to go ahead and add our next reagent. And here we have our ethanol. This is really high strength, like 100% ethanol. And so we add a little bit of it to each tube. And what that does is it what's called precipitates out the DNA. And so that makes the DNA fall out in a solid form rather than being a liquid. So we consult our protocol and it tells us to add 200 microliters, the same amount. So use the same pipette, use the same tips. And we take our ethanol, we'll grab the whole rack, grab our 200 microliters of ethanol, and go ahead and add it to our sample. And there we go. Now we do that for the rest of them. Right. So ethanol has been added, and just about after every step here, after we add anything, we go ahead and mix by inversion again. So there we go. We're going to do this 20 times. 3. All right. So now we're going to move from these tubes to our filter tubes. Then we get to go ahead and centrifuge them, which is the fun part. So for this, we're just pulling the whole volume out of the original tube and putting it into the correctly labeled collection and filter tube. So for that, it doesn't, as the filter columns can hold up to 600 microliters on them. So we're going to set this to 600, and this should pull out just about everything that's in the tube. And there's that. And there it is, added to our filter column. And it doesn't leak through because it's a really tight filter. If you notice, there's a hole on the bottom, but the only way to get the liquid through the filter is by using a centrifuge and spinning it really, really fast. So that's what we'll be doing here in a sec once I finish transferring all of these. All right, so that's done. Those are lids. Those tubes are done. Now we load them into the centrifuge. So there's the lid, just in case anything does go flying, it hits the lid and not anything else. So pull the lid off, and we load our tubes into the wells. And the important thing to do when loading a centrifuge is to evenly space apart all the tubes. So if you were to put all these together, it'd be like throwing something like a brick into a dryer or a washing machine. You'd hear it going boom, boom, boom. And at the speeds these spin, that can be really dangerous. So it will definitely damage the centrifuge, and it'll throw it off balance, so it won't spin as well. But this is a small centrifuge. We have a bigger one in the lab that's probably, the center part's 2 to 3 times the size. And that at full speed, if it were to become horribly unbalanced, parts of it would come flying, and that would be really bad for anybody standing next to it. So we're going to spin it for 1 minute at 8,000 RPM. So that means each tube would make 8,000 revolutions in a minute, which it's going to do. So we have our setting on the front. It's pretty straightforward. Set it to 8, set the time to 1, and we go ahead and make sure the top is closed and click start. So when those come through, we're going to have in the bottom, in the collection tube portion, right, not the filter, is going to be mostly junk. So we added the ethanol, and that precipitated out the DNA, which made it solid, right? And so the solid's going to get stuck on that filter on the top. And so that's good, but everything else that goes through is going to be junk that we don't want. So that's going to go into a beaker, which will then be placed into hazardous waste. Then we're going to keep the top, so the DNA is going to be on the top. There's going to be a few other things with the DNA that you can't see, and so we have 2 more wash buffers to run through the column. And then once it's fully washed, then we have this thing called Elution buffer, which will make the DNA into liquid again and let us collect the final volume of DNA. So those go there. We pull our lid off, and we pull out our freshly spun tubes. So if you saw going in, the liquid was in the top, and now it's in the bottom. So we're going to take that, dump the liquid out into our waste vessel, and since we still have more junk to wash through, we're going to just use the same collection tube. At the very end, when we're collecting the DNA after the addition of the elution buffer, we'll use a new collection tube that doesn't have any residual, because you dump most of it and there's always a little bit of liquid left in. All right, so we dumped everything, close that again so nothing falls in there. And we move on to our next wash buffer. So, like I said, we're cleaning off the DNA, and so we're going to add 500 microliters of this guy into each column, and then we're going to centrifuge it again. All right, so the first wash buffer has been added. Now we go ahead and load it back into our centrifuge. And making sure every time we load our centrifuge that it's balanced. So we're going to read ahead while this spins. So the flow through, the stuff that ends up in the collection tube, is still going to be waste in this one. So what we're going to do is the same thing we did with the last one. It's just take it out, dump it. And then once we've dumped it, we'll be adding the second wash buffer called AW2. The first wash buffer removes proteins we don't want. The second wash buffer removes salts. So proteins are a lot bigger, salts are real small, just individual molecules. Now this time, since we're removing, mainly just small things that you can see, that's why it looks like mostly water. Like, oh, you don't see anything there. Well, unfortunately, you can't see most of the things we work with. You aren't going to visually see the long chains of DNA. So, for now, we just have to believe that they're in there. But at the end of this, we'll be using a machine called a spectrometer that lets us see how much DNA is in there, and also helps let us check if there are any contaminants. All right, and this is going to look like a repeat of the last step because it mainly is. It's just with a different wash buffer. So, all right, our second wash buffer has been added. So for the second wash buffer, because it's dealing with smaller stuff, it's better to let it sit for just a little bit. So we're going to go ahead and load it into the centrifuge, but we'll give it a minute or two before we actually start spinning it. That way it lets it soak onto the filter and really dissolve all those salts that we're trying to get off of the DNA. If we don't properly clean the DNA, it'll interfere with a lot of the things we do later with the DNA. So you'll be seeing more about the PCRs with Lisa. And so this is going to be our last wash buffer. And so we're trying to get any liquid on the columns off of them. So for this one, we're going to turn the speed up to maximum. So for this centrifuge, that goes up to about 14.8 thousand RPMs. And we're going to spin it for 3 minutes so that all the liquid is definitely off. We're done with these collection tubes. So the flow through is mostly clear, and our old collection tubes are contaminated with all the liquid that was in them. even though you could dry this out, it'd still be there. So the whole thing gets to go into the waste container. And this guy, which you can't see it, but there should be precipitated DNA still on the top of the filter. So he's going to go into a brand new collection tube that came fresh out of the bag, is clean and sterile and doesn't have anything in it. All right. So last step, last little bit of liquid to add is the elution buffer. So how much elution buffer you add decides how much final volume product you'll have. So for this, because we're dealing with just a small amount of DNA for the most part, and the tissue samples we used were pretty small, so we're going to use only about 50 to 100 microliters of elution buffer. I'm going to go with 100 just because I've done this enough times that I know 100 will be enough for what we need. You don't need a whole lot of volume. It's better to have a higher concentration in it. So the more volume you add, the more total product you have, but it's more dilute. More pipetting, same as before. So the elution buffer, because of the way it works, it has to sit right on top of the filter for how many minutes? 5 minutes? 5 minutes. So I'll add it, we'll wait 5 minutes, and then we'll go ahead and centrifuge it. Just make sure that all the DNA that's on the filter gets fully dissolved. That's been added. Check our watch and wait for 5 minutes. All right, so our samples have incubated for 5 minutes, which is just a fancy word for incubate at room temp, just means let them sit. So if you stick the things in the incubator, then it controls the temperature and it's warm in there, which helps the digestion process. But for this, we just have to let it sit and soak. All right, so this is the last spin in our centrifuge. We're going to let it spin for 2 minutes at 8,000 RPM. And now this spins for 2 minutes. So when this is done, we're going to go ahead and toss the filter columns off the top. There won't be anything in them we want anymore, and you can't reuse them because they're contaminated with, just DNA. So the top part will get tossed now, and we'll be keeping the bottom, and we'll be aliquoting the bottom into these little tubes for longer term storage. And usually when we do this, we split our sample when it's done into multiple. So it's got the same number, but just a different letter. And that's just, if anything happens to this tube, we still have the second one archived. All right, all done with this guy. We can close it and turn it off. And then we'll go ahead and what's called aliquot our sample or our DNA product into their tubes. Usually we'll only toss one filter column at a time and let the rest sit. That's to make sure anything in the air doesn't get into our sample. And two, the only labeling on the tubes is on the top. So once you pull them all off, they all look the same. So that would be bad because we don't want to mix them up. All right, that one's done. All right, so there's our DNA extraction. So after this, these get thrown into a very, very cold freezer, usually at minus 80 degrees Celsius, just to make sure that they stay stable. DNA sitting at room temperature will eventually degrade and break down. We're going to go ahead and spec these on our spectrophotometer, and we'll see how pure the DNA is and help quantify it roughly. For our next step, we get to look at our samples on this guy, our BioSpec Nano. It's a spectrophotometer, and basically it's a big fancy word broken down: spectro for spectrum, photo for light, so it's a light spectrum meter. DNA has a couple unique properties when you shine light through it, and because of the molecules and the chemicals that make up DNA, some of them absorb light, and they absorb light at a very specific frequency. So we have 260, 280, 260, 230. And so what this machine does is it shines the light through the sample that we'll put it onto this tiny dot here, and it lets us know how much of the light made it through, and knowing that, we can have a pretty good idea of how much DNA is in that sample. And also it gives us a nice graph of how much light made it through and at what frequency. So we can also see if there's contaminants or salt that's left over or things like that in the DNA. So the amount of liquid we put on this is 3 microliters, which is going to be a very, very small amount. The first thing to do is we have our elution buffer here from our last step. So this is what the DNA is in. So what we have to do first is we set what's called a blank. So the meter can kind of adjust or ignore certain things. And so since we definitely know it's in the AE buffer, we will use the AE buffer as our blank. So we take 3 microliters and we load it onto this real tiny speck here. Which can be kind of tricky. But you do it. And then you click this button up here that says blank, and you can hear it clicking softly, although I don't know if that's going to make it through the mic. But so the blank set, there won't be anything up on the screen just yet. We punch in our sample name, 24009, since that's the number we're doing first. And we grab our pipette tip and our sample. And take our 3 microliters of what's hopefully a good bit of DNA. And then you cross your fingers. And we get this nice curve. So the amount of nucleic acid concentration, so that's what the NA in DNA stands for, is listed here. And that's a very healthy concentration. That's pretty great. So that makes me happy. Normally with this curve, if I washed the DNA a little bit better or a little bit longer, you'll see a deeper valley here, and then it'll come back up for a peak. So that sample, this is fine for running PCRs, and so we'll go ahead and check our next sample. Punch in the sample name, grab your sample. Now this one should be interesting. Yeah. So this is what is, although the number's lower, there's less DNA in it. This valley's deeper, so this is a cleaner piece of DNA. So this is all the DNA of the slug. If you take a piece of DNA from any part of the slug, you'll get its full genome. And same thing with us, same thing with just about everything. You can do a simple mouth swab and you'll collect a whole bunch of DNA and probably some bacteria DNA and other things too. So what we do with these slugs is you can't look at a slug and tell whether or not it has rat lungworm. So that's why we do a lot of this. Well, part of this is we take the slug that we've trapped and we digest it and we get all the DNA, including if there was rat lungworm in that slug, there's rat lungworm DNA in here. So the next step after this is the PCR.